Technical Veterinary Learning Channel, honored by Prof.Ahmed Mamdouh El-Sherif, Professor of Epidemiology and Infectious Diseases, the ex-head of Department of Veterinary Medicine, the ex-Vice Dean of Faculty of Veterinary Medicine, Bani Suef University, Egypt, presents today. African horse sickness disease will be explained, clarified in several series. African horse sickness has another synonyms such as Peridisceti, Pistis equorum, Lapisti equina, Pistis equina africana, and in Sudan its synonym is Abu el-Manazir. African horse sickness molecular pathogenesis. Incubation periods of African horse sickness. There are both types of infection. In natural infections, circumstantial evidence indicated that incubation period ranged from 7 to 14 days, while experimentally, African horse sickness usually showing 5 to 7 days incubation period, by means that experimentally the incubation period half the times. This is maybe due to dose of the virus is directly injected into the experimental animals. Incubation period will depend upon the virus load, the way of infection, and the animal health status, which reflected on its immunity level. And according to forms, epidemiologists and clinicians found that there are different in days for incubation period experimentally as the following. Pulmonary or per-acute form, incubation period taken about 3 to 5 days. Cardiac or odometus or the subacute form, incubation period ranged between 7 to 14 days. In acute or the mixed form, the incubation period taken 5 to 7 days, while the African horse sickness fever taken the 5 to 14 days as incubation period. For understanding the African horse sickness molecular pathogenesis, we have to understand the ultrastructure of African horse sickness virion. So, in the following, we will discuss and clarify the schematic presentation of the ultrastructure of the African horse sickness virion is non-enveloped with a triple capsid structure and is about 18 nanometer in diameter, enclosing the genome and transcription complexes. The genome of African horse sickness virus is composed of 10 double-stranded RNA segments of linear coding for 12 proteins. They encoding 7 structure proteins from VP1 up to VP7, which sequenced for African horse sickness virus serotypes 4, 6, and 9. 4 non-structured proteins from non-structured protein 1 up to non-structured protein 3A. This is the diagram that describes what I said. In this second diagram, we noted that the inner core layer has T equal 1 symmetry with each of the 60 units composed of the homodimer of VP3 or virus protein 3. Proteins VP3 and VP7 are the major inner capsid proteins. Proteins VP1, VP4, VP6 constitute minor inner capsid proteins. Non-structure 3 protein are the second most variable African horse sickness virus proteins. Most variable being the major outer capsid protein and VP2, which is the determinant of African horse sickness virus serotypes. VP2 with VP5 are target for virus neutralization activity. This diagram showing that the outer core of the virion is composed of proteins VP2 and VP5 with 260 trimers of VB7 and has T equal to 13 equihedral symmetry. The outer capsid layer consists of 120 globular trimers of VP5 and 60 triskelion shaped spikes of VP2. The most old abbreviated pathogenesis of African horse sickness virus. This pathogenesis is still used to clear the pathogenesis of African horse sickness up to September 2019. This diagram showing that African horse sickness virus enter by insect bite, and the virus if go to the wall of RPCs, it remain for 1 to 3 months, masked from antibodies, causing the mild form. While if the virus go to the vascular endothelium, it will replicate, and this leads to increase in capillary permeability. This increase in capillary permeability leads to hemorrhage, edema in the head, neck, thorax, and lungs, and those leads to occurrence of acute form and subacute form and mixed form. According to later old diagram of pathogenesis, the pathogenesis of the virus is not fully understood because of pathogenicity and virulence studies of African horse sickness virus are difficult to perform in horses for logistical, ethical, and financial reasons until September 2019, where post that date complete clearance of the pathogenesis had been established, as will we clarify below. Outcome of infection is highly dependent on viral factors, as studies denoted to presence of certain proteins that are linked to virulence of the virus inoculum. This diagram explains and clarifies the pathogenesis of African horse sickness virus. African horse sickness virus enter the cell by attachment of viral protein 2 to sialic acid receptors on the cell wall and either clarithin-mediated endocytosis or microbinocytosis. Acidic pH in the endosome causes loss of viral protein 2 and mediates viral protein 5 membrane pre-mebalization which results in uncoating of the virion and release of transcriptally active core particle into the host cell cytoplasm. Transcription and translation of viral proteins occur utilizing the host cell machinery, and the viral IBS act as, which means that viral internal binary action, which act as sites of assembly for the progeny of the virion. Assembled core particles are then trafficked from VIP on the exocytic vesicles by non-structural protein 3 interaction with clabitine. The outer capsid proteins VP5 and VP2 are acquired during this process to produce mature virions. Particles are released from the cell via budding mediated by non-structural protein 3 or via host cell lysis. The whole process will be more clarified with continue of the video. African horse sickness virus host infection is believed to be initiated by the outer capsid protein VP2, host cellular surface receptor, and capsid protein VP5, which contains a characteristic coiled coil motif typical of membrane fusion proteins. Due to the location of putative salicylic acid SA binding domain in VP2 trimer hub, have postulated that VP2 trimers may attach to the surface of cells through two different interactions. The first one, antigenic tip domains, bind to certain surface cell receptors which have yet to be identified. There is some preliminary evidence implementing heparin sulfate as one possible candidate, but due to high sequence variability in the African horse sickness virus outer protein VP2, it is possible that different receptors and entry mechanisms may be utilized by the different serotypes 4, 6, and 9 to enter distinct cell types. These initial bonds are then stabilized by a second connection between the SA or putative salicylic acid binding domain and a surface glycoprotein, such as glycopherrin A, which is a heavily glycosylated sialoglycoprotein abundantly present on the surface of equine erythrocytes. Sensitivity of virus protein 2 to serum protease has been established, and it has been suggested that a central domain at the top of African horse sickness virus protein 2 triskeleton hub, could be the target site for such a horse serum protease. This domain is situated directly cleavage of African horse sickness viral protein 2 in this region would thus increase accessibility to this potential binding site. These are GD sites on VP7 protein may play a role in the ability of viral clefts to infect cells. The process is believed to be initiated by proteolytic cleavage of viral protein 2, either in infected insect saliva or in the host serum, post which the virion attached to the host cell membrane. These initial studies suggested that viral cell entry was accomplished by clarithin-mediated endocytosis. However, more recently evidence has been presented which describe a micropintocytosis like entry route dependent on actin and dynamine. In both instances, the low pH within 6 up to 6.5 within the early endosome distribute the interactions between VP2 or viral protein 2 and viral protein 7, well facilitating detachment of the viral protein 2 tremors and distributing the zinc finger motif situated at the interface between viral protein 2 hub and the body domains, which is believed to play a role in controlling conformational changes. Together with a further lowering of pH in the late endosome, the removal of viral P2 causes a refolding of VP5, which in turn leads to outward protrusion of prop-like structure from particle surface with viral protein 5 tremors remaining tethered to the particle by their anchoring domains. These harp-like fusion peptides insert themselves into the endosomal membrane of the cell of the host, causing release of viral core particle into the cytoplasm, which may play a role in the ability of viral CLPS to infect cells. These harp-like fusion peptides insert themselves into endosomal membrane, causing release of viral core particle into the cytoplasm. Removal of the outer capsid proteins and the release of viral cores into the cellular environment containing necessary host substrates and transcription factors causes the core to become transcriptionally active machine for production of new virion. Each of gene segments is then simultaneously and repeatedly transcripted by viral protein 1 to produce ssRNAs, which are modified by capping and methylation activity of enzyme of the viral protein 4 within the core before being released into the cytoplasm again. Viral dsRNA is thus kept within the core particle, protected from detection by component of the original host cell innate immunity. The nascent ssRNAs act as messenger RNAs for the synthesis of viral proteins using the host cell machinery, and later on in the newly formed cores are templates for dsRNA gene synthesis. The VIPS or viral internal binding act as the sites of viral assembly and protein non-structure to play a role in directly and specifically sequencing the 10 ssRNAs together with the 3 enzymatic protein, viral protein 1, viral protein 4, viral protein 6, and inner core protein, viral protein 3, for encapsulation and the formation of new subcore particle of the virus of African horse sickness. Deposition of viral protein 7 trimers serves to stabilize particles, and phosphorylation of non-structural protein 2 then regulates their exit from the virion internal binding in order to acquire two outer capsid protein VP5 and VP2 for formulation of mature progeny virions. Although release of these virions from infected mammalian cells is predominantly affected by cell lysis accompanied by significant cytopathic effects, viruses have also been shown to use budding mechanism for viral egress earlier on infection cycle. All latter processes explained is mediated by utilizing host exocytosis pathway and membrane destabilizing action of non-structural glycoprotein non-structure 3, which functions as a viroporine and also interacts with calaptin to function as a bridging molecule between new virion and host cell export machinery. In insect vector, where establishment of a persistent viral infection is important, there is no observable cytopathic effect, and viral release is mediated excessively via vessels formation at cytoplasmic membrane. It is interesting to note that more variable African horse sickness non-structural protein 3 causes a much greater cytopathic effect earlier on the infection cycle, possibly indicating that this protein is expressed at higher level or it is more toxic. Another experiment model that done in April 2019 pathological characterization of African horse sickness virus infection in a mice and naive horse addressed some questions about the true pathogenesis, experimental infection of interferon alpha receptor chain, and also naive horse with a strain of African horse sickness virus serotype 4, then collected data about clinical signs, histopathology, immunohistochemical analysis of immune response to infection, antigen detection in tissues, and transmission electron microscopy of African horse sickness infected tissues. This will be explained in the rest of this video. Results indicated that African horse sickness virus serotype 4 infection is correlated with edema and pneumonia in the lungs, inflammation in the liver, and meningitis, plus prevascular coughing in the cerebrum. Other data shows that different strains of African horse sickness virus differ in terms of their pathogenicity and tropism. Horses which recover from African horse sickness virus exhibit solid humoral immunity against homologous challenge. Protective antibodies appear to be directed toward neutralization epitopes on African horse sickness virus viral protein 2. Different strains of African horse sickness virus was induced in 5 naive horses by inoculation for determination the ultrastructural changes in the endothelial cells of capillaries in myocardium, lung, spleen, and liver. Equidi developed both cardiac and mixed form of the disease. Alterations detected in endothelial cells of the vessels of infected animal included presence of structures associated with viral infection, hypertrophy, degenerative changes, appearance of cytoplasmic projections, a change in permeability, alteration in intercellular junctions, loss of endothelium, subendothelial deposition of cell debris and fibrin, as well as vascular repair. In association with these changes, edema, hemorrhages, and microthrombosis were detected particularly in the myocardium and lungs. This experiment proved that infection of and changes to capillary endothelial cells of organs was independent of the disease form in which the disease manifested itself, but was dependent on the organ itself and also the blood vessel type. Thus, different levels of African horse sickness viral tropism were observed for different endothelial cells of vessels in different organs. Viral infection was commonest in endothelial cells of microcardial vessels, followed by endothelial cells of lung, whereas in spleen and liver, the endothelial cells infection was rare, as well as in the liver, the viral infection will be limited to interstitial capillaries. From the clarifying of the ultrastructure of the virion and the new theory of pathogenesis that linked to this ultrastructure, we can know the accurate pathogenesis of African horse sickness virus in details. This detail tells us how the viral components make occupation and control of the host cells leads to all forms of subacute, acute, mixed, and chronic form of this disease. African horse sickness series to be continued. Please subscribe the channel in order to be able to access the latest information and explanations.